Hello, class. I hope all is well. This chapter, the nervous system, is a very long chapter. I'd like to give you a few introduction as far as what's going on. It's a very long chapter. It's a very important chapter, I think. And what I would like to do, I would like to break it down to two. We've done it before. I don't want to talk for an hour and a half to get bored. I know anytime you like to pause it and do whatever you want to do with it. But I like to break it down to two sections. And it is, most of you in here are psychology major. So this is a very important chapter for all of you guys in here. And I hope you get the most out of it. Very important chapter. Anyhow, it's probably one of the most important chapter that we studied, the nervous system. Again, a lot of your psychology major, if not any other major, I think it's very important. But anyhow, let's start. So these are learning outcomes. A nervous system receives a process by definition. I try to read this chapter. I try to read and elaborate, and sometimes say, I'll already talk about it. But anyhow, information from both external and internal environment. So the light you're receiving, the taste you're tasting, the touch, all of that external. And then sometimes internal environment, you have an upset stomach because something did not go physiologically, something inside did not go very well, something is happening. So that's what the nervous system respond to it. So you are coordinated, you're controlled by two things, your nervous system and your endocrine system. Those two control and coordinate you. And then the endocrine system is going to come up, that's your glands, okay? It's going to come up. That is also an important chapter. Those of you who are psychology major, it would be good to study that chapter a little bit more in depth. Anyhow, sensory information from both external and internal environment. Internal environment, we're talking about inside of you. What are some of the stuff that goes inside of you, your nervous system controls? It has two major divisions, central nervous system, CNS they call it, or PNS, peripheral nervous system. Central nervous system is your brain and your spinal cord. Peripheral nervous system, anything in your arms, your body, your legs, what have you, that is your peripheral nervous system, which we will talk about it. Here they are, they broke it down to your brain and also to your spinal cord, which is your central nervous system. Then the peripheral nervous system, all of the arms that goes to your arms, legs, your abdomen, your chest, everything else outside of brain and spinal cord is called peripheral. Periphery, we've heard of periphery around this room. It's a peripheral nervous system. And this is another thing about this chapter. The PowerPoints are great. That whoever done the PowerPoints for this chapter did an excellent job. Whoever wrote the chapter did an excellent job. It's easy to understand, easy to read, and I hope I explain it the way your chapter has done it. So anyhow, then of course, as I talked about it, your central nervous system is broken down to two parts, brain and A and B, if you would. A, if you're easier, or 1 and 2, whatever you want to, your brain and spinal cord. Then these two are in touch with each other. They talk to each other. It's not like they're two separate entities. It's continuous. Your brain is continuous with your spinal spinal cord. Then what happens? The brain, the central nervous system, it has the information from your sensory neuron or afferent neuron from the environment or from inside of your body, goes into brain and spinal cord, okay? And then the sensory neuron, as your textbook broke it down, is somatic sensory nerve signals from skin, from muscles, joints, spatial senses, which there are 5 senses. We will talk about it in the next chapter. And then also your visceral sensory nerves, your signals from body organs, all of that send information, sensory, afferent, away if you would, away from the organ in trouble, okay, or not in trouble necessarily. And it goes to the brain, and that neuron, and I will show you a picture of a neuron later on. Hang on, okay? A neuron is a nerve cell, okay? From that neuron, it goes to the brain and the spinal cord, they analyze it, do whatever they have to do, and then they send the information back to motor neuron or efferent neuron. So the information, after they analyze it, they deal with it, they send it to motor neuron, which carries information from CNS, central nervous system. This is all peripheral nervous system now, remember? And then it goes into somatic motor signals, nerves, and then it goes to autonomic motor nerves. Autonomic nerves are broken down to sympathetic and parasympathetic. So as you can guess, the rest of the chapter is pretty much this diagram, okay? That's what the rest of this chapter is about. And then I will talk about that a little bit more. Okay, the overview of the nervous system, the sensory. Okay, I didn't talk, there is something between the sensory neuron and motor neuron. It's called interneuron or association neuron. So between the two, there is another neuron, if you would, my black marker here, that is association neuron, black, right? Okay, so it's called integration, and there is a name for, the two names I have for that, association neuron or interneuron, it will come up. So I already talked about sensory neurons. They send the information to central nervous system. Motor, just like a motor engine, okay, just spelled the same way. Motor pronounced same way. The motor neuron will take the information from central nervous system to the tissues, to the skeletal muscle, smooth muscle, glands, any organ in the body, okay, that they have to. And then in between, as I said, the central nervous system sums up the input it receives from all over the body, stores and memories, and creates motor responses. It's called integration. And then sometimes there is an interneuron between them, which I will go over that. We'll talk about it. This is what is called, it says the neurons will regenerate themselves. That is true when you cut yourself here or there. Not necessarily always, all the time, your spinal cord is cut, it cannot regenerate, but that's a big nerve cord. Sometimes you cut a nerve cord in your arm and then your sensations are limited. But these are, we're just talking about a neuron. Just think of it, a cut in your arm. So what happens, you don't lose the sensation here. It's just a cut, you cut the neuron is cut like here. So what will happen, it will regenerate itself. And I will talk about Schwann cells in a minute, just hang on. And then after it regenerates itself, the sensation goes back to your hand and everything is gonna be normal, okay? So that's what that power is saying. Nervous tissue, so contains the tissue. You remember I talked about four different types of tissue at the beginning of semester. We have epithelial tissue, nervous tissue, connective tissue, and muscular tissue. Remember that? We talked about those. Now let's go back and revisit nervous tissue. It contains two types of cells, okay? And you know the definition of tissue, combination of cells together perform specific functions. So I kind of like to watch my time too, okay? So there are two types of nervous tissue. One is neuron and the other one is nuclei. Nuclei, as the neurons transmits impulses, of course, information is sent from one neuron to the next neuron. That's how it is. And then nuclei, they are supportive and nourish neuron, okay? They are supportive cells, okay? I had one student a long time ago, and he said, Amir, the nuclei, some of them also transmit information, just like neurons to some extent. Okay, so, but we will stick with not most recent research, but we'll stick with what your textbook is talking about right now. Greatly outnumbered neurons, nuclei in the central nervous system. They are broken down to 3 types, your textbook: microglia, astrocytes, and oligodendrocytes. Nuclei in the PNS is Schwann cell, okay, PNS, peripheral nervous system, central nervous system, 1, 2, 3, okay, and there's only one in the peripheral nervous system, the supportive cells, okay, so we're talking about supportive cells. Okay, the nervous tissue, macroglia, there he's talking about these 3 are CNS, right, and this one is PNS. Let's see what they have, your textbook has to say. Macroglia phagocytizes cells that remove bacteria and debris, just like your white blood cells. Okay, they act like your white blood cells. But they are in the central nervous system, in the brain and spinal cord. Astrocytes look like a star, as the name appears, look like a star. Astrocytes provide metabolic and structural supports directly to the neurons. Okay, and oligodendrocytes forms myelin sheath in central nervous system. So they are the ones that makes the myelin sheath, and I will talk about that. Very important concept, very important muscular dystrophy, and we'll talk about it. Hang on. And then Schwann cells form myelin sheath around the PNS. Okay, and again, you will see that. 3 types of neuron based on the function: sensory neuron, interneurons. That's what I was talking about. Another name for interneuron is association neuron. Okay, so another name for it is association neuron, same thing. Okay, and motor neurons. Sensory neurons carry nerve signals and sensory receptors, detects changes in the environment and so on and so forth. 3 types of neurons: continued interneuron, found only within the CNS, okay, in the brain and spinal cord. I'll give you one example, which is reflex arc. When you put your hand on a hot stove, you quickly remove it. That's an example I will elaborate on, and we'll talk about interneuron there. Hang on. I think that's going to be in the second video. Receives inputs from sensory neurons and other interneurons, sum up signals received from other neurons, then communicate with a motor neuron. Motor neurons carries the nerve impulses, already talked about that, to the effectors. It could be muscle, organs, or glands, which carries out their response to the environment change. Okay, so anatomy of a neuron, let's talk about that, what the neuron, a nerve cell look like, and what is it, what are the components of it. The components of it are 1, cell body, 2, dendrites, 3, axon. Okay, just the neuron. The cell body contains the nucleus and other organelles within the cytoplasm. The dendrites are the extensions of the cell body that receive signals from the environment. Okay, and then an axon is the portion that extends. You can remember it. Axon extends of an extension of a neuron that it goes and sends the nerve impulse. Individual axons are termed nerve fibers, and collectively they are form a nerve. Okay, right here, here is the axon. You guys can see that. Here is a cell body. Here is a sensory receptors in this case. And then here are the axon terminals right here. For this neuron, this association neuron or interneuron, that was the sensory neuron, right? For this association neuron, these are all dendrites, all of these. This is cell body, the nucleus inside of it, cell body, nucleus, and other organelles. So nucleus and cytoplasm is called cell body. And then this is the axon. And it ends up, look at it, it ends up into the next neuron, which is the motor neuron, right here. The motor neuron. And then of course, these are all dendrites, like a branch of a tree, and then cell body, and then the axon right here. And at the end of the axon, these are axon terminals right here, like a doorknob. And then the effectors would be right here. That would be your skeletal muscle. That would be a gland. That could be a organ, anything that we already talked about it. Okay, myelin sheath. I said I'll give you a rain check. Here it is. I'm cashing out on the rain check that I gave you. It covers the some axons and form when Schwann cells, PNS, or oligodendrites wrap around the axon many times, and node of Ranvier space between the myelin sheath. I gotta show you pictures. Myelin sheath, again, quickly, gray matter. He's talking about gray matter, but you already know, and white matter, you already know what cell body is. You already know what the axon with Schwann cells wrap around them. We'll talk about that. Myelin sheath, the myelin sheath is important in nerve regeneration within the PNS. I'm waiting for a picture. Physiology of a neuron, nerve signals, electrochemical changes that convey information within the nerve. Maybe I should go back to resting potential. Yeah, I gotta go back. I'm sorry, guys. This is not, okay. Maybe it's right here. And I will talk about it. Let's talk about myelin sheath. So here is the axon, right? Remember that? And then you see the blue part right here, that there is one cell right here, there is another cell right here, another cell 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 here. So there's 1, 2, 3, 4, 5, 6, 7 cells are here. If you look at it, that blue thick layer, that is made up of phospholipid right here. You guys can see that, okay? So the phospholipid, if you would, right here, that is the cell membrane, like take a pizza dough and wrap it around your arm several times. And that would be a myelin sheath, okay? These cells, these Schwann cells also have, they do have, the Schwann cells also have nucleus, they have cell body, but think of it this way. Here is a nucleus and cell body and everything else, and right here is a big extensions of cell membrane. So that big extension of cell membrane wraps around the neuron. Okay, I hope I'm making some sense. And right here, if you would, and they're showing it to you guys right here as well. All neuron have Schwann cells, but not all neuron have myelin sheath. So imagine if a neuron does not have myelin sheath, take a pita bread or a tortilla and wrap it on your arm, right? That is a Schwann cell, wrap it on your arm, and it does not have myelin sheath. But if you take a pizza dough and wrap it on your arm, it goes around a couple of times. A huge, large pizza, extra large pizza. Okay, it goes around your arm a couple of times, 2, 3 times, 4 times. And that's myelinated. That's myelinated Schwann cell. Okay, so all neurons are surrounded, all axons are surrounded by. Yes, they're showing you a picture of a myelin sheath. This is a transmission electron micrograph. Look at the myelin sheath from all here to here. Okay, and this is the axon. All of this is the axon. Okay, they cut it in a cross section, so you're looking at it like this. Okay, they cut it in cross section. So I did talk about it. So myelin sheath covers the some axons, and then a node of Ranvier. So right here is a node of Ranvier. The junction between that is very important. You do need it. Ranvier was a French physiologist, anatomist that discovered it, medical doctor. Right here, these are node of Ranvier between Schwann cell and Schwann cell. Between Schwann cell and Schwann cell, these are node of Ranvier, and you can see it right here as well. Okay, so node of Ranvier spaces between the myelin and axon. Myelin sheath continued, so gray matter, white matter, it will come up. So remember, the gray matter contains no myelin sheath, is all the axon. And if if you cut up chicken or meat, anytime you see the fat kind of creamish, okay, this yellowish color. That's what they call it. The very first people who cut into brain and spinal cord, they say, wow, this is gray and this is white. Okay, they didn't call it black and white. They call it gray because it is gray. And without staining, they had no clue what is going on at that time when they. So they call it gray matter, white matter. And later on, of course, they learn, oh, gray matter has the cell bodies, and white matter is the myelin sheath, which is fat. Okay, myelin sheath, important nerve regeneration. I think we talked about that. Nerve signaling. Okay, physiology. Now we pretty much talked about the anatomy of a neuron, histology of a neuron. But let's move on to physiology, the function of a neuron. So the resting potential, I need to talk about that. I need to talk about polarized neuron. Very good. A picture like this. Okay, so what is resting potential? There is a lot of sodium outside of sodium and potassium. There's mainly a lot of sodium outside of the neuron right here. There's a lot of sodium and potassium outside of a neuron. And what will happen, reference records electrodes, it's a reference, okay. And then if you put an voltmeter, this is a voltmeter right here, and you put one electrode out of it, one electrode inside of it and one electrode outside, and you turn on the voltmeter, you see minus 70 millivolt. It means inside of the neuron is minus 70 millivolt compared to outside of neuron. So inside of neuron, they have these molecules such as protein molecules, ATP, they're negatively charged right here. So inside of neuron is more negatively charged than outside of the neuron. Resting potential, sodium outside the axon, potassium and large anions inside the axon, okay? So inside the axon, you have a lot of potassium right here. I'm getting my pictures mixed up. So I guess yes, okay, I got it. Now I know what's going on. This is, I thought that is outside. This is outside, this is inside, and this is outside. So there's a lot of sodium. I'm gonna write it down, so no mistakes. There are lots of sodium outside of a neuron, okay? And there is some sodium inside, and the cell is pumping it out, constantly pumping it out by a sodium-potassium pump, which we studied it a little bit before, okay? That is called resting potential. It means a neuron is relaxed. It doesn't do anything. Just relax. That's resting potential. When there is more sodium ion outside than inside. When you attach a voltmeter, the voltmeter shows that inside of neuron compared to outside, that's potential. That's a term, polarized. A membrane, the axon, this is the axon membrane right here. They draw it like that, okay? And it's long. Remember that? You saw it in the pictures before. That membrane is outside of the phospholipid, outside is, okay. So resting potential, continuum potassium pump, sodium potassium pump, I talked about that. There are 3 sodium out and 2 potassium in when we talk about sodium potassium out. So what happens when a neuron is poked, you stimulate a neuron, what will happen? That's called stimulus. Stimulus is a change that activates the neuron. Right now I can feel myself if I poke in a classroom session. I used to poke the students once like this, so you can feel it. And then when you, that's a stimulus. So the stimulus has to be strong enough to go through a threshold. Threshold is a level that the neuron can be stimulated. If it does, the stimulus is not strong enough. The stimulus right here is not strong enough. I'm touching myself, lying a little bit, but if the stimulus is not strong enough, you cannot feel it. So action potential will not occur. Here it is. So what will happen? This is a threshold potential. So remember, this was, this is inside, right? I forgot. Yes, this is inside, this is in, and this is out of the neuron, right? So a lot of sodium went in, a lot of sodium went in. So when the sodium went in, the threshold is minus 55 millivolt. It means if the neuron does not reach that minus 55 millivolt, you will not recognize anything. Even though it was stimulated, but it will not recognize anything. So from minus 70 millivolt, it went to 54, 55. So action potential is all or none. So what does that mean? All or none. If this portion of the neuron is stimulated, there's action potential in here, and neuron is long. Remember, the sciatic nerve goes all the way from your back to your leg, all the way to the bottom of your leg. Remember that? Okay. So it's long, very long. If it is stimulated, it means all of the neurons stimulated. It's just not this segment is stimulated. That's what all or none means. All of that 1 meter, or in some people a little bit longer than 1 meter. So that's what a all or none means. Depolarization, it means that sodium cannot stay inside of the neuron. It has to come out. It will come out. Okay, that's depolarization. When the action potential begins, protein channels open up, sodium rushes in, that's depolarization. Repolarization means all of that sodium got to come out. That's depolarization. I skipped 1 slide. Sorry about that. So depolarization is when the sodium rushes in. You're depolarized. Remember, the outside and inside. Right here, finally, it reaches the action potential is plus 35 millivolt. It reaches that so much sodium gets in. This is in again. This is out. So much sodium comes in that inside of the axon becomes positively charged. How much? Plus 35 millivolt. Very small voltage, but still. Okay. Depolarization continues, sodium gates open up and move inside of the axon. Repolarization, I was jumping in ahead. Sorry about that. It means all of that sodium must come out. Get out of. Immediately after depolarization, sodium channels close and potassium channels open up. So the potassium flows out of the cell and inside of the cell becomes negative again. Okay. Finally, the sodium potassium pump. There we go. 3 sodium, 2 potassium, 3 sodium, 2 potassium. These pumps take him out. Here it is, and goes back to resting potential, repolarization. Repolarizes itself. Okay, it has to go back to minus 17 millivolt. Here's a nice graph. And then, of course, here is a minus 70 millivolt. This is the threshold, 55. It has to be stimulated. The stimulus has to be that strong that many sodium gets in. When that little bit of sodium gets in and the stimulus is strong, then a lot of sodium gets in and it gets 35 plus 35, roughly 35. And then, of course, right here, potassium moves to outside the axon and sodium potassium pumps move the sodium out. This period of repolarization, this time, remember, from here to here is about 3 milliseconds. Very fast, 3 milliseconds, 3 to 4 milliseconds. Very fast. But this period right here, about 1 or 2, 1.5 milliseconds, is called refractory period. It means no matter how strong of stimulus I give myself, the neuron will not respond. Okay, it will respond immediately here, right here. You can have another action potential right around here. But from that time to that time right here, of course, refractory period, if you take physiology, They will talk about their different absolute refractory period and partial refractory. They talk about that a lot more. But don't worry about it for now, for your class. Okay. Propagation of action potential, I talked about it. It is all or none. And then the neuron that they have, unmyelinated neuron, you have Schwann cells around them. But if you have an axon like this, and then you have Schwann cells like that, right? Okay. And here is the axon terminal. Here is a cell body, right? You guys know what I'm talking about. You know that. So what happens, the propagation is all or none, of course. If you have the myelin sheath, these are the myelin sheath around it, the action potential jumps from one node of Ranvier to the next node of Ranvier to the next node of Ranvier. That is faster, since it jumps, action potential jumps from one node of, that is faster than a axon, that is faster than the axon that does not have. So the action potential must go through all of this until it reaches the end. Here, since it jumps, it's faster. Okay. It's called saltatory conduction. Okay, propagation of action potential continued. If myelin action potential, saltatory conduction, I talked about that, make sure you study that. And right here, the action potential is jumping from one node of Ranvier to the next node of Ranvier. Remember, I talked about this. This is called node of Ranvier. Okay, so it jumps from one node of Ranvier to the node of Ranvier. Refractory period, I talked about that. Immediately after the action potential, during which axon is unable to conduct another action potential. This ensures the one-way direction, so it goes through and so on. Synapse is a space between two neuron axon branches, and to find axon endings called axon terminals, like a doorknob. Some textbook they call it axon knobs, axon terminals. Each terminal lies very closely with the dendrite or the cell body of the another neuron. Okay, this region is close proximity is called synapse. So quickly, when I draw it, here's you have a cell body. Here is an axon, an axon terminal, axon knob right here. So there is another axon right here, which is dendrites right here. Another axon. I hope you guys know what I'm talking about. Okay, so that space between these two, that space right here, very small space, is called synapse. Okay, I hope you know what I'm talking about. Synaptic cleft, the small gap that separates the sending neuron from the receiving neuron. Neurotransmitters are the little molecules that will go ahead, stimulate, and I will talk about it, transmit action potential across the synapse and stored in synaptic vesicles in the axon terminals. And I will talk about that, I guess, if the picture comes up. The nerve signals travel along an axon and reaches an axon terminal. Calcium ions enter the terminal to stimulate the synaptic vesicles. Yes, neurotransmitters released into the synaptic cleft and diffuse across the receiving membrane. There, okay, here we go. Great pictures and pictures of pictures. So, all of these PowerPoints that we talked about it. Here is the synapse, right? This area. Okay. This is the axon knob, axon terminal of the presynaptic neuron, they call it. And this is the postsynaptic neuron, right here on this diagram. Okay, so synaptic cleft, they call it synaptic cleft. So what happens, these are the axon, these are the vesicles, sorry about that. These are the vesicles, they have neurotransmitters. What happens, calcium must come inside of the axon knob for the vesicles to be formed, to be moved. Okay, so these vesicles, with help of these calciums, move to here. And then when they bind with this phospholipid of the axon knob, axon terminal, and release the neurotransmitters here. The postsynaptic neuron, the receiving neuron, the postsynaptic neuron have these blue dots, you can see that. and they attach to the receptors of the post neuron and then the receiving neuron or receiving glands. And then what happens? Sodium, your good old sodium, gets in because of these neurotransmitters. Right here, they draw it for you, sodium. Okay. Without these neurotransmitters, sodium would not get in. Without action potential, these vesicles would not release neurotransmitters to the next. So now to the next neuron. So now you know the importance of action potential when it's poked. When it's poked, then action potential goes across the axon and then reaches the axon now and releases the neurotransmitter. Neurotransmitters open up the sodium channels for the postsynaptic neurons. So action potential goes from one neuron, the message goes from one neuron to the next neuron. I hope I'm making some sense. Removal of neurotransmitter, yes, those neurotransmitters cannot sit there. There is an ACE at the end of the word. It means enzyme. Acetylcholinesterase, ACH is the abbreviation for the neurotransmitter, and E at the end, it means an enzyme. Acetylcholinesterase, there are enzymes right here on postsynaptic or receiving neuron, and they break down acetylcholine to acetyl plus choline. Before here, they're together. But when they reach that enzyme, acetylcholinesterase, it breaks it down to acetyl and choline. So they are deactive now. Removal of the neurotransmitter contained, the existence of the neurotransmitter. So I talked about all of that. Whatever extra, please read it and make sure you know it. Again, I don't want to talk too much. I have a meeting in about 15 minutes. I gotta go in there. Yeah, acetylcholine, norepinephrine, dopamine, serotonin, glutamine, GABA, all neurotransmitters. Okay, and they all have different functions. Do not get them mixed up. Okay. As the chapter goes on, we will talk about some of them. Nerve organs and glands, synapses also use a neurotransmitter. Acetylcholine and norepinephrine are active in both CNS and PNS, which you will see it a little bit later on. Neurotransmitter molecules, serotonin, and so on and so forth. Please read them. Make sure what are the functions. Serotonin is involved in the thermal regulations, body temperature, sleeping, emotions, perceptions, and many drugs influence them. Integration, something of, I talked about that a little bit before, so I'm gonna let you guys read it. So when there are, the neurons are stimulated by inhibitory, they call it, and stimulatory neurons. Okay, so inhibitory synapses and excitatory synapses, they release both type of neurotransmitters. He's talking about that. The most important stimulatory, excitatory signal neurotransmitter is acetylcholine, and the most important inhibitory signal is GABA. Okay. So let's see if I can, I like to talk about a little bit, just a little bit about the brain, and then we stop. I'm not going to go into great detail. So let me go ahead at least break you into it and talk about it a little bit. Okay, so the central nervous system, he's going into parts. Okay, the meninges are right here. The meninges are made up of epithelial tissue which surrounds the entire nervous system all the way from brain to the end of spinal cord. And then, of course, meningitis is, God forbid, is a bad disease. Kids, if they get it, it can cause death in kids. That's why they say anytime you have a child less than a month old and they have fever, immediately take him to a doctor, take him to a hospital. And the first thing comes to the doctor's mind is meningitis at that early. I'm different. I'm a little bit different than doctors. But anyhow, it can cause death in kids. But anyhow, cerebrospinal fluid. So there is a fluid that percolates throughout our central nervous system, all the way from brain to spinal cord. It goes down and it goes between meninges and pushing there, protects the central nervous system and central canal and ventricles. You will see central canals. If you have spinal cord right here in between, there is a hole and that canal. If it moves, the spinal fluid moves there, and it is painful sometimes for some diagnostic problems, or when women are giving birth, they go into the spinal tap instead of spinal tap. They go in there and they give medication, or they draw some from there to check for HIV or some diseases. And but anyhow, they don't go to ventricles as far as I know, but they go to central canal and they withdraw spinal fluid. Cerebrospinal fluid normally exists of however, blockage can cause problems. Anyhow, hydrocephalus is when the brain is full of water, and that's not good. Here are the ventricles of the brain where the water can percolate, lateral ventricles, okay, third ventricle, fourth ventricle, and spinal cord. Right here, that's central canal, which the fluid, these are where the fluid of the brain percolates and go around. And of course, the idea is to get rid of, if there is a disease or bacteria or something, and it percolates and the system can get rid of that unwanted pathogen. The central nervous system is composed of two types of neurons, of the nervous tissue, gray matter and white matter. I talked about those. And spinal cord extends from base of the brain through the foramen magnum. You remember that in the skull? That was last chapter. The biggest foramen in your body, foramen magnum. That's what they call it, magnum huge. It's on the bottom of your skull, so the spinal cord can pass through. Structure of spinal cord, a cross section of spinal cord is shown, shows a central canal, gray matter and white matter. You do have a slide of that for the lab portion of this course. The gray matter is like a butterfly, right? And then right in between, you can see the central canal, and white matter surrounds it. And gray matter is where the cell body are. And the white matter is where the myelin sheath and the axons are. Okay, so spinal projects from the cord, small openings, intervertebral foramina, okay, intervertebral discs, and you already talked about that. That's what we have in the lab. This is your gray matter. And right here, that's your central canal. Okay, they're showing you. And the fluid percolates through there. And they're showing you, this is a nice diagram. They're showing you the spinal cord, which is surrounded by your vertebrae. It's being protected. Okay, so your brain by your cranium or your skull, and then your vertebrae are protecting your spinal cord. Well, a centrally located gray matter, as you saw that dorsal root of the spinal nerve contains sensory fibers entering the gray matter. The ventral root of the spinal nerve contains motor, and so on and so forth. The ventral side, that's where your mouth is. Okay, the dorsal side, that's where your back is. So this is the ventral side of our body. So on the ventral side, he's talking about that you do have these dorsal root ganglion raised. Okay, the ventral here, ventral root ganglion, okay, and this is dorsal root ganglion right here. So in the back. So when you look at your spinal cord, you have the front ventral and the back dorsal. That's pretty much those terms mean. Front, back. Okay, structure of spinal cord concluded, white matter. I'll let you guys read it, many tracks. I talked about it, that the axon is cut, it can regenerate. But what happens if the spinal cord is cut? Big, huge nerve bundles, if they're cut, they cannot regenerate. Okay, scientists are working on it for centuries. So paraplegia is when is damaged at the thoracic region of the spinal cord, the lower body and legs are paralyzed. Okay, paraplegia. Para, it means paralyzed. Okay, quadro, it means four. Quadriplegia is damage if the neck region right here and all four limbs are affected. Okay, so now you know the difference between quadriplegia and paraplegia. And I would like you to know that for exam and paralysis, patients in accidents. Of course, there are stories that people came back and they were able to walk again, but pretty much. Reflex arc, okay, function of the spinal cord continued. When you put your hand, let me talk about it quickly right now. I do not have a picture, but he's talking about reflex arcs. When you put your hand on a hot stove, you quickly remove it even without thinking about it. So what happens, the sensory neuron send the information to spinal cord. Spinal cord does not send the information to the brain, okay? The information goes through an association neuron or interneuron, and it goes back to motor neuron. And the motor neuron says, hey, move the muscle so you don't burn yourself more, okay? That's what a reflex arc is. Another example of reflex arc is when you go to a doctor and they put a hammer in your knee jerks. That's a reflex arc, okay? So the information does not immediately go to the brain. Later on, it goes to the brain. It goes to the spinal cord and comes back. The doctors put an flashlight into your eye, so see if the iris of your eye enlarges or closes or not. That's a reflex arc. These are all three reflex arcs that I'm familiar with. Okay, brain is made up of four parts. So we are moving from spinal cord to brain, guys, now. Four parts of the brain: cerebrum, which is the largest portion of the brain, diencephalon, cerebellum, and brainstem. Cerebrum, let's talk about the 1, 2, 3, 4. Okay, A, B, C, D. So the cerebellum is called telencephalon. In embryonic development, telen, it's the tela, it means you have it in your prefixes, suffixes, end, finished. Okay, so during the embryonic development, largest portion of the brain, and it has a lot of curvatures, communicates with coordinated activities of other parts of the brain. Here it is. So this is a great diagram. This is possibly the best thing I've seen. They did not put a lot of details. They just put whatever you need to know for beginners. So appreciate this diagram. So cerebrum is this whole entire, all of this is, I'm coloring it a little bit. All of this is cerebrum. Okay, that's it. That's the largest portion of your brain. And then of course your brain has two halves and they are connected together by corpus callosum right here. Okay, he's showing you the meninges, the protective layer outside of the brain. Then diencephalon right here, which I will talk about it a little bit. It's made up of thalamus, surrounded by third ventricles, and hypothalamus and pituitary gland. So A, B, C, 3 parts make up diencephalon, midbrain. Okay, brainstem is made up of the midbrain. Sorry, brainstem is made up of midbrain, right, A, if you would, pons, B, and medulla oblongata, which continues to spinal cord, that's C. So brainstem, midbrain, pons, and medulla oblongata. And cerebellum is right here, the second largest portion of the brain, cerebellum. Okay. And these are just, he's showing you the ventricles, spinal cord. They are not part of cerebellum. They're not part of the cerebellum. Okay. Anytime he's showing you ventricles or other portions, he did a great job. I would not put these here because they are not part of cerebellum. Anyhow, students can confuse it. Okay, I did label them for you. And the brain is made up of two hemispheres, right and left. Okay, and then those big folds like here, that's called gyrus, singular, gyri, plural, and then these indentation, those valleys, if you would, not this one. These valleys are called sulcus, sulci, plural, sulcus, sulci, okay? Cerebral hemisphere, longitudinal fissures, a deep groove that divides the left and right. Oh, right here. That's a longitudinal fissures. That's a fissure right here. I said, this is not a sulcus. Remember that? That's your fissure. Okay. So that is your fissure, deep groove. And corpus callosum, I talked about it, communicate the two halves, extensive bridges of the nerve tracts, and gyri and sulcus, I talked about that, the folds and sulci are little grooves in there. Your brain has 4 lobes, which we need to study it. Anterior frontal lobe, which is right here, parietal lobe right here, occipital lobe in the back, and then 2 temporal lobes right here. Okay, and as you can guess, temporal lobe are helping with hearing, occipital sight, frontal smell, and then of course parietal is involved with a lot of things right here. So it gives you primary somatosensory area, parietal, and then right here he's telling you the functions of each lobe. Frontal, primary motor area, motor, you all know what motor we talked about it. Premotor area, motor speech, and Broca's area, prefrontal area, and so on and so forth. Sense of smell should be here. Olfactory, sense of smell. Olfactory should be here as well. Temporal, so on and so forth. I'll let you enjoy it. Occipital, visual, primary visual area, right here. Make sure you know those. Cerebral cortex, thin, highly convoluted area out of the gray matter, responsible for sensation, so on and so forth. We talked about it. Primary motor area, central sulcus, and again, I'll let you enjoy and read. And there's not much I can explain. Here they are, a nice diagram showing you. You don't have to know this diagram, first of all, for your exam, your quizzes. But it tells you that your parietal lobe is responsible for all of these functions, if you need to know that. Primary somatosensory area, again, primary taste, visual, auditory. I talked about those a little bit in the parietal lobe, occipital lobe, temporal lobe, and frontal lobe. Okay. The association neuron, promoter area, somatosensory association neuron, I think visual area, auditory area, again, repeat of those. Wernicke's area, the posterior part of the left temporal, Broca's left frontal lobe, and then it gives you the functions. Basal nuclei, Parkinson's disease, degeneration of the specific neurons in basal area, which diencephalon. Okay, let's stop right now so I go to my other meetings as well. But I will come back and talk about diencephalon, which is made up of A, hypothalamus, B, thalamus, and pineal gland, C. And then we'll talk about that. All right, guys, I'm gonna stop.